Hi guys, welcome back to another lesson. This is module 1, biology, part 2, cell functions. So the inquiry question is, how do cells coordinate activities within their internal environment and the external environment? Okay, moving on. First, we're going to talk about the transport of materials. So, let's talk about solutions first. A solution is formed when a solute, when it dissolves in a solvent. The amount of solute dissolved in a given quantity of solvent, that determines the concentration. A very concentrated solution has a lot of solute in relation to the solvent, and a dilute one has a very small amount of solute. Okay, so moving on, the movement of material. So basically, there are two kinds of movements. The first one is passively, and this doesn't need energy. The molecules, they're moving with the concentration gradient, which is high to low. The second one is actively, and this requires energy because they're moving against the concentration gradient, which is low to high. Okay, so let's talk about diffusion now. Diffusion is the movement of molecules, except for water, that's an exception, from a region of a high to a low concentration. And it does this until equilibrium is reached. Equilibrium is basically when there's no net movement of molecules in any direction. All of the molecules move equally in each direction. This process does not require energy. It's passive because it moves with the concentration gradient. Diffusion is a pretty slow process, not gonna lie. The rate is affected by 1. the concentration gradient, 2. particle size, 3. temperature. So basically, the particle size, the smaller the particles, the faster the rate of diffusion. And why is this? Basically, this is because when you have small particles, their surface area and the amount of energy needed to travel is actually slower. So yeah, if you guys don't get this, I'm gonna highlight it so you guys can refer to this later. The third point is temperature. So the higher the temperature, the higher the rate of diffusion. This is because when you increase the temperature, you're basically increasing the energy, which means you're also increasing the kinetic energy, which means there's a higher rate of diffusion. This is because it causes the molecules to move faster, yeah. Anyways, the difference in concentration of a substance in two areas, it will be divided by a barrier, and that will be called the concentration gradient. The rate of diffusion changes depending on the concentration gradient. So as you can see in this area, there's a lot of solute here and not a lot of solute here. So all the solute, like the molecules and the particles, will move this way. Yeah. A large difference in the concentration means steeper concentration gradient, and the diffusion will occur faster. A small difference in the concentration means that there will be a gentler concentration gradient and diffusion will occur slower. Now let's go on to osmosis. Osmosis is it's like a special type of diffusion. Basically, it's not the movements of anything else. It's the movement of water molecules only from a region of high water concentration to a region of low water concentration. It does this through a selectively permeable membrane. This process doesn't require energy. That means that it's passive. So osmosis is passive. As you can see in this diagram here, so all the water, it's concentrated here and it's not concentrated here, so it's just like moving that way. Yeah. Now let's actually apply osmosis. So basically, water moves into and out of the cell membrane through osmosis. It doesn't move directly through the lipid bilayer, but it moves through a special protein channel called aquaporins. Basically, these are just water pores. When the water concentration outside of the cell is higher, which means there's low cellular concentration. And this, it might get you like mixed up. Basically, you just gotta think of it the opposite way. So when the water concentration outside of the cell is higher, which means it's low cellular concentration, than inside the cell, which is high cellular concentration, water will enter the cell by osmosis, causing the cell to swell and possibly rupture. This rupturing is called cytolysis. When the water concentration outside of the cell is lower than inside the cell, water will leave the cell by osmosis, causing the cell to shrink, which is called plasmolysis. So this is a hypertonic solution, this is an isotonic solution, and this is a hypertonic solution. Now these names, they might get very confused with. You're just gonna have to remember them. So basically, as you can see here, this is normal, this is flaccid, and this is plasmolyzed. And this one, it's a hypertonic solution, and it's shriveled because there was less water concentration outside of the cell, which means it left the cell, which means it's shriveled. And this one, hypotonic, there was too much water. There was too much water surrounding the cell that it moved in, and it ruptured. It swelled. Yeah, so that's a graph. Osmosis in a graph. An increase in the mass indicates that the solution is hypotonic. An increase is hypotonic. A decrease means it's hypertonic. 0.0% change means it's isotonic. So basically, hypo, increase, hyper, decrease. Okay, so let's think of an acronym. So O and ER. O, increase, ER, decrease. Now moving on to the exchange of materials. The following factors will affect the exchange of materials across the cell membrane. Chemical characteristics of materials such as chemical factors and physical factors, and the concentration and the surface to volume ratio This is very important, and it's usually written like this. Characteristics of materials, chemical factors. So basically, uncharged particles can easily diffuse through the cell membrane because they're like easily dissolved because they're uncharged. However, charged particles such as sodium ions, they require protein channels to travel through the cell membrane. Water is not lipid. Therefore, it moves into and out of the cell through aquaporins. We talked about this earlier. Physical factors. Size and shape affect the movement of substances across the cell membrane. Yeah, small molecules can diffuse easily, but large molecules, they require a protein channel. And very large molecules will be transported into the cell by endotosis or exotosis. We will talk about this later. Concentration gradient. The relative concentration of the substances on either side of the membrane affects the rate of diffusion. High concentration means it will diffuse rapidly because the concentration is higher. Decrease, which means it's a slower rate. In order, I don't know what happened here. Anyways, facilitated diffusion. Large molecules and charged particles, they cannot diffuse through the cell membrane. Oh my god. Anyways, large molecules and charged particles cannot diffuse through the cell membrane. They are transported into or out via the proteins. There are two types of proteins, channel proteins and carrier proteins. This process is called facilitated diffusion. Each protein acts on the membrane transporter. Is specific to one solute or several similar solutes. This allows the movement of a large or charged molecules to channel proteins. Small ions such as sodium ions will diffuse through the cell membrane via protein channels like this, a narrow passageway that spans the cell membrane. This works like gates that open and close. Yeah. Carrier proteins. They bind molecules on one side of the cell membrane and then they change shape and release the molecules on the other side. So it's like you go in there and then it's like a specific shape for it, and then it will change shape and release to the other side. Now we're going to talk about the second type of transport, which is active transport, and it requires energy. So do y'all remember like what active transport was? Yeah, it's the area of low concentration to an area of high concentration. And this is against the concentration energy. And it requires the input of energy. So as you can see here, it's low concentration and it's going to a high concentration. Active transport requires a carrier protein that spans the membrane to actively move chemicals from a low to a high concentration. This is utilizing cellular energy. One example is when glucose and amino acids are reabsorbed by kidney cells, so they are not lost in urine, or when the end or when the end products of digestion are loaded from the intestines into the bloodstream. This is to absorb food. Transport of large molecules. So this is talking about, so basically particles that are too large to move through the cell membrane by either diffusion or active protein transport can be transported actively by endocytosis. Do y'all remember that term? When a large particle, a large particle has to be moved into the cell membrane will change shape to surround and engulf it. This process is called endocytosis. If it's a solid, it's called a phago. If a liquid, it's called a pinocytosis. Cell drinking and cell eating. Like, as you can see, the membrane, it like surrounds the food, and then it will Now, time for exocytosis. Cells produce many waste products because they're living, and these wastes are removed through the process of exocytosis. During exocytosis, a membrane-bound vesicle moves to the cell membrane, fuses with it, and then releases its contents into the fluid outside of the cell. The vesicle becomes part of the cell membrane, so basically it merges into the cell membrane and then it's released. Like this part, it will just be like that. Anyways, this is a summary table, and next we're going to talk about cell requirements. First off is a basic summary of surface area and volume. Surface area is the outside area of the cell. It's the number of faces times height times length, but you won't be able to measure it in an exam. Volume of the cell is the space taken up by the internal contents of the cell. So basically, volume is the height times the length times the width. Surface area to volume ratio. The surface area it's divided by the volume is called this thing. So this ratio affects the movement of substances into and out of the cell. The cell needs to have enough surface area to the volume to meet its requirements and remove the weights and to take in the things as well. A small cell has more surface area in relation to its volume. So as the cell grows, the ratio decreases. Therefore, large cells have a smaller amount of surface area. It is have a low SAV. Yes, so next we'll talk about the transport of materials based on this ratio. The distance from the surface of the cell to its center is much smaller in a smaller cell compared to a larger cell. Therefore, the smaller size allows a faster movement of substances between the center and the surface. Oh my god! Okay, the distance from the surface of the cell to its center is much smaller in a smaller cell compared to a larger cell. I think we know this from above. So if a smaller size allows faster movement of substances, anything that the cell needs to get all the parts of the small cell quickly and waste can be removed easily. This allows the cell to perform at an optimum level of functioning. Yes, so as you can see here, if cells get too big, the cells will not be maintained its homeostasis and it will die because they need a good SA to volume ratio to maintain good health. So the cell grows too big, it will divide cell division into two cells so it can maintain that volume ratio. Cell requirements. All organisms on Earth use matter to produce the energy required for biologic processes. Organic compounds are synthesized by autotrophs and are made up of these atoms. These include carbohydrates, lipids, proteins, and nucleic acids. Inorganic compounds are part of the non-living world and do not contain this in their long chains. These include these. Anyways, moving on, carbohydrates. Carbohydrates are the basic subunits of carbohydrates are simple sugars called monosaccharides. Y'all are just gonna have to remember this. And disaccharides, two sugars joined together, and polysaccharides. So basically this just means one, this just means two. You just gotta remember saccharides and you'll know. And polygon, yeah, many sides. So basically, carbohydrates, they're important because they're like an energy source. Glucose is an monosaccharide and it's used for quick energy. Starch is a polysaccharide and it's used for stored energy. It's also useful for structural components. Cellulose is a polysaccharide and is used for strength and support in cell walls. Yes. So you're just so basically you don't have to, I'm pretty sure you don't have to remember that. Just remember mono is quick, poly for stored, and these two have to remember it's for structural and energy sources. Lipids. Basically, lipids are composed of a glycerol head and a fatty acid tail. They are arranged in a triglyceride as glycerol molecules are attached to three fatty acid tails. In cells, lipids have two main functions: energy storage, fats and oils, and structural component of membranes, a bit like carbohydrates. Moving on, let's talk to proteins. Amino acids are basically the building blocks of proteins, and amino acids, they join together by peptide bonds, and these form polypeptides. One or more polypeptide chains, they will twist together and form a protein, and these, they have to form in a specific shape as well. So functions of the proteins include: they will form structural components in the cell's membrane and tissues, and some even have a functional role. Enzymes control metabolic processes. Hormones control the functioning of cells. So here's a summary. Amino acids, they're linked by this, and they form this, and yeah. Nucleic acids. Nucleic acids carry the genetic information of cells. There are two types of nucleic acids, DNA and RNA. This is a double-stranded molecule that stores the information that controls the cell. Basically like an instruction manual for the cell. RNA is a single-stranded molecule that assists in the manufacture of proteins. Basically, this is a diagram. That is a nucleotide. This is an RNA, so it doesn't have this part of it. And this is DNA, and it's like, yeah. Moving on, this is a summary table for inorganic compounds. Removal of cellular waste. Not all the products that are produced from chemical reactions are required by the organism. These products are called waste, and they have to be removed so they won't like accumulate and will interfere with how the cell functions. The removal of these wastes is called excretion. Oxygen and carbon dioxide, they're usually the waste products from a chemical reaction of photosynthesis and cellular respiration, respectively. So basically, oxygen is photosynthesis and carbon dioxide is cellular respiration. These gases can be removed easily from the cell by diffusion through the cell membrane. We talked about this earlier. Excess water, it's sometimes not required by the cell and moves out of the cell by osmosis. We also talked about this. Lysosomes, lysosomes contain digestive enzymes and are responsible for breaking down and removing old cells and other cellular wastes. Any of the waste that can't be eliminated by the lysosomes, for example, hormones and enzymes, they are packaged and removed by exocytosis. We also talked about this. Basically, it's the membrane and how Metabolism. It's a total sum of all chemical reactions occurring within a living organism. Each step of the metabolic pathway in a cell is catalyzed by enzymes. Metabolism is divided into two categories, anabolistic and catabolic. Anabolistic is building up large organic compounds from simpler molecules. These actually require an input of energy. So these are some examples. Catabolic involves breaking down into simpler ones, and these will give out energy. So ana and kata breaks, ana forms. These are some examples. Enzymes are proteins, and a protein is a long chain of, we did this before. Enzymes, they're biological catalysts. They control the rate of a chemical reaction. An animal cell, they have 4,000 different types of enzymes, each catalyzing a different chemical reaction. Every type of enzyme with a specific shape has a specific shape, as it is made up of a specific pattern of amino acids, which is basically proteins. Within their structures, enzymes have active sites that are usually composed of 3 or more amino acids. The active sites are areas that substrates the molecules that in this thing will act on. So basically what it's the reactants that the enzyme will act on will bind to catalyze chemical reactions. When an enzyme binds to a subject, it will make a new molecule called the enzyme substrate complex. I know it does sound a bit complex, but it will get easier. As you can see, the substrate, it binds to the enzyme to create this thing. And this is a more complicated figure. Active sites, and this is the enzyme, and this is the substrate. So basically, the role of enzymes, they are to speed up reactions without a change in temperature. In chemical reactions that occur, heat could be provided, the necessary energy, but in the living world, heat burns tissue, so that's obviously not a way to go. For a chemical reaction to begin, activation energy is necessary. The role of an enzyme is to lower the activation energy needed to start by bringing specific molecules together rather than relying on them colliding randomly, so that the reaction can be proceed quickly without a change in temperature. This is good. So lowering the activation. Just check. Okay, guys, we're nearly done. We literally have like 10 pages left. For a chemical reaction to take places, the molecules involved, they need to collide at the correct orientation and with the right amount of energy. This is called activation energy. So as you can see, the uncatalyzed, it's a lot of energy, but if you catalyze it, it's very less. So enzyme models, there are two models to describe the enzyme specificity. These are the lock and key model and the induced fit model. Lock and key model. It was originally thought that the enzyme's active site was rigid and a small molecule, and it's reciprocally shapes and fits into the active site, like how a lock fits into a key. Once the enzyme substrate has formed, the close proximity of the molecules allow the reaction to be rapidly catalyzed and the products to be released. Note how the enzyme remains unchanged after the reaction has taken place. They are only needed in small amounts and can be used over and over again. These actions are always reversible. So as you can see, this is like the lock, that's the key, it binds and then it breaks, like look how, and then it doesn't even change. Second model is the induced fit model. The induced theory assumes that the subject plays a role in determining the final shape of the enzyme substrate complex, and the active site is more flexible. As the substrate enters in and binds to the enzyme, shaping the active site and properly aligning the enzyme for the reaction to take place. Other substrates may fit in the active site, but unless they are properly able to shape the enzyme, the reaction will not be catalyzed. So as you can see, that the active site's like very weird, and this is like a rigid shape. When it goes in, it will change shape to suit, and then it will catalyze the reaction, and at the end it just goes back to its original state. So in this thing, it doesn't change at all, the enzyme. Coenzyme. Many enzymes, they cannot function without a coenzyme, also known as a cofactor. And a coenzyme is a non-protein group that binds with the protein parts and helps form the active site. It increases size of the enzyme molecule and its active site, increasing the chances of collision. It can be easily separated from the protein parts, but its presence cannot function without the cofactor. A functional enzyme may therefore consist of the protein only, or it may be in the form of an enzyme cofactor complex, where the enzyme part of the complex is the protein. So this is basically what it looks like. It will look like that. Like if it fills in there, it will not work, so it needs a coenzyme to like be there to like make the shape more proper. Characteristics of enzymes. Enzymes, due to their protein nature, they are sensitive to temperature, pH, and substrate concentration. They are temperature sensitive. Oh my god. They are in they function normally around 37 degrees, which is around body temperature. Anything above or below this temperature, their efficiency decreases. At temperatures above, most enzymes will stop functioning altogether. The enzymes will break down. The structure and shape will start to change in a process called denaturing. We will go through this. Denaturing of an enzyme renders it useless because the substrate cannot bind with the active site. It will no longer be able to function properly to help catalyze the reaction. Once denatured, the change is permanent. The enzyme cannot be reformed back into its original shape. See, as you can see here, it denatured and it can't fit back in. Yeah. What's the difference between? Oh, okay. Yeah. They're also not like excessive cold. It will also cause it to slow it down or stop functioning. But however, cold is not permanent. When the temperature is increasing, the enzyme will start functioning again. This is not denaturing, guys. You gotta remember this. So this is the optimum temperature. It's also really sensitive to pH. Each enzyme has a region of pH optimal stability, which varies greatly, depending on the reaction catalyzed by the enzyme. So, like, when you're in the stomach and the enzymes need to catalyze stuff, the stomach is, like, very acidic. So, obviously, they need, like, an acidic thing. pH, yeah. Acidic and basic are two extremes that describe a chemical property of substances. Mixing acids and bases can cancel out or neutralize their extreme effects. A substance that is neither is neutral. pH is a way to describe the acidity. The pH measures how acidic or it's basically a measure of the hydrogen ion concentration. pH scale ranges from 0 to 14. I don't know why it's so... There you go. Anyways, 7 is neutral. Anything less than 7, acidic. Anything greater than 7, basic. Oh my god, I'm so done with life right now. Oh my god. Anyways, pH sensitive. So the enzymes are pH sensitive. The shape of the graph is usually symmetrical on either side of these like peaks. The optimum pH, the enzyme's 3D shape, is ideal for the substrate. So the reaction is at the pH lower or higher than the optimum, the enzyme's shape will begin to change. The substrate will no longer fit, so the activity is less. At extreme pH, the enzyme can be denatured, and that is not good. Substrate sensitive. Enzyme molecules are specific, acting on only one type of substrate. Therefore, each enzyme catalyzes one particular chemical reaction involving the substrate for which it is specific. The rate of enzyme-controlled reaction is affected by the concentration of this means that the amount of the things that the enzyme has to catalyze beyond certain substrate concentrations, the rate of reaction is limited by the amount of enzyme. Rate of reaction. Enzymes are very efficient. They work rapidly, having a high rate of reaction. For example, the rate of the action, so in the absence of an enzyme, this reaction is very slow, only 200. In living cells, this reaction is catalyzed by enzyme this and this much. Like, that is a huge number, formed every second, and that is per hour. Can you believe it? That's actually crazy, bruh. Is substrate sensitive. As the substrate increases, the rate of reaction will also increase because more enzyme is available to catalyze the reaction. At higher substrate concentrations, the rate of reaction will plateau because no more enzyme is available to catalyze the reaction. So basically, it will become flat. The maximum rate at which the reaction will occur is called the saturation point. So basically, you can see here, it's like less. There's a good amount, but there's too much. There's too much, not enough enzymes, too much concentration. Adding more enzymes. At a saturation point, if more enzymes are added, the ratio would once again increase, but then it would level off because it would become saturated again. Here is your enzyme summary. I'm gonna log off now. My voice is going to die. Thank you, and remember to subscribe. Yeah, bye.